start off this morning's webinar and the November meeting for Auckland Beekeepers Club. Thank you very much indeed for those who've dialed in. We've got a good lineup this morning. So just briefly, we will be having first Dr Hendrickson from Massey University, a lecturer and microbiologist and specialising in bacteriophages. So these are basically bugs and germs which can feed on other things which might be unwanted in a beehive or other environments. Then we'll have a recorded presentation from Ken Brown on using the club's extractors and how to use them, hire them, practical demonstration, that type of thing. I'll then be talking a bit about supering up for the honey flow, for the nectar flow, which is running quite well at the moment. And then another talk from Ken on lighting a smoker and how to keep it going while you're doing the hive inspections and so forth. Just briefly, a couple of bits and pieces about the club. Hopefully, and fingers crossed, this will be our last webinar. We had wanted to make Heather's talk an in-person one, but of course we're still operating under level 3 lockdown, even of step 2, and therefore it's a webinar instead of in-person. But we expect at the end of this month, given the indications that the Prime Minister has been giving so far, that we'll be switching over to the new traffic light system, and this will enable us to meet in person. So at this stage, the club is planning that our December meeting, which is the 11th of December, will be an in-person meeting, and that we will do this as our end-of-year barbecue. I thought just really important after so many months of being apart and away and having to do things over webinar. I thought the first priority for us, let's just get together as a club, stand round with a sausage and so forth and drink in hand and catch up and tell stories about our terrible COVID here and the indignities that we've had to go through over the past months and just generally catch up as a club and enjoy each other's company again. So at this stage, that's our plan for December meeting. December the 11th will be our end of year barbecue. So put a ring around that on your calendar now and look forward to catching up with everybody and meeting you again after an enforced period apart. I hope you and your beehives are all going well. As I say, nectar flow is going at the moment. So we've got some information coming up in later talks relevant to this time of year, as we always do. The club application into the council is still proceeding in terms of our new home and where we'll be moving to. So hopefully we will have more to report on come our December meeting. I think that's just about it at the moment. Oh yes, the journal has been posted out. Some of you may have already received it. If not, it will be arriving within the next day or two. And there's some very good articles and interesting things in there. So have a good read of that, and also the program for the next month's coming up, of course, as well. So I look forward to seeing all of you. In the meantime, I'm afraid you're going to have to watch us on screen yet again. But I'd particularly like to welcome Dr. Heather Hendrickson. And Heather, if you can take over and talk to us about bacteriophages. After your interview that you had with Ken last month was fascinating, and particularly looking forward to listening to what you have to say. All yours. Thank you.
Great. Thank you so much, Steve. And thank you to Ken and Dave for organizing and for the lovely chocolates that showed up at my door this morning. It's really a pleasure to be invited, and I'm looking forward to telling you guys a bit about what we've been doing in our project with bacteriophages, and thinking about bacteriophages and how they can help the honeybee industry, and particularly with the problem of American foul brood. So I'm going to share my screen so that you can see my talk. So the project that I'm talking to you about today is the ABATE project. And the ABATE project is something that we started maybe 3 years ago. And now we're looking to expand the project and kind of open it up into some new directions. And it's really all about using bacteriophages to produce, to further support the health of hives. So first, I'd like to start by just saying that, you know, the work that I'm talking about today is primarily really done by the brave postgraduate students in my laboratory at Massey University. So I'll point out the students that did the work as we go. And also, I need to say right off the bat that without the support of the Howard Hughes Medical Institute's training and education project, we really wouldn't have started the Massey Phage Fauno, and which is the group of faculty, phages, and students that are involved in phage hunting at Massey University. And we've gotten a lot of really generous support, both from AgMart and from the Sustainable Farming Fund. And I should mention also the New Zealand Honey Trust most recently. So we've had a lot of support, and there are a lot of other people that are involved in this work. So the first thing that I'd like to do is just introduce you to a bacteriophage. And I'll show you some real photographs of bacteriophages later, but basically this is what bacteriophages look like for the most part. So they have a capsid that's made out of protein, and they have a tail, and they have these little connectors, which are part of their tail that allow them to identify the bacteria that they infect. And they're incredibly numerous and absolutely everywhere. So you can find bacteriophages in the milk in your refrigerator, in your honey, in your skin, in your gut, in the soil around us, in the oceans. Bacteriophages are 10 times as numerous as bacteria, which are already incredibly numerous. So they're extremely common, and they are everywhere. And it's been over 106 years since we first discovered these bacteriophages, which really means kind of eaters of bacteria. And so what do they do for a living? So a bacteriophage is really a protein particle that floats around in its environment, kind of bumps into things. And as it does that, if it recognizes its specific host, it is able to inject its DNA into that host. And the DNA is able to take over the replication machinery inside the cell and make as many as between 10 up to 200 copies of that bacteriophage in as little as 20 minutes. Once those are assembled and inside the cell, they burst out of the cell. And when they do that, they destroy that bacterial cell. And this is the way that they commonly operate, and it's called a lytic cycle or lysis. So then each one of those individual bacteriophages is able to go on and infect more of that same type of cell. And so when this was first discovered, it was actually before we discovered the incredible power of antibiotics. And because of that, today, if you take an antibiotic like meropenem and you put it in droplets on a bacterial lawn, on a plate, on an agar plate, you can see clearings, and you can see this is the evidence that bacteria are being killed by that antibiotic. And similarly with bacteriophages, once you find them, if you drip them onto a similar type of agar plate, you actually can see that the bacteriophages kill the bacteria. So somebody has just asked, and I'm kind of keeping my eye on the Q&A, but I might miss some of these. So a bacteriophage is a virus. A bacteriophage is a virus that specifically infects bacteria instead of infecting something else like us or bats or anything else that you might be thinking about, particularly in our coronavirus age. So these are viruses, and they're everywhere, and they kill bacteria. All right, and because they have this power to kill bacteria, they've actually been used as medicine in Georgia and Poland and Russia, and I've actually visited a bacteriophage factory in Poland where they were producing bacteriophages for industries like the poultry industry to kill bacterial pathogens in poultry. So this is a tool that's being used in some parts of the world, but it hasn't had a lot of use until quite recently in Western medicine. But very recently, we've seen the rise of stories like Stephanie Strathy's story of bacteriophages being used to actually save her husband's life when he was infected with Acinetobacter baumannii. So this is a recent book, I think it came out 2 years ago, where Stephanie tells the story of what happened to her husband and how American scientists that had discovered bacteriophages were able to take those bacteriophages and administer them to her husband, Tom, and actually save his life. It's a great story. This is also, bacteriophages have also been approved for use on food. And here in New Zealand, we even have one phage product that's used in the beef industry in order to destroy E. coli. So these are not incredibly, not used all the time, and something that might be a little bit unfamiliar, but they are gaining in popularity and in use because they have this incredible power. So some of the advantages of using bacteriophages against bacterial pathogens is that they, when you take an antibiotic, sometimes that will have negative effects on all of the bacteria in your system, which is why sometimes we get side effects from that involve losing some of the beneficial bacteria in our guts when we take antibiotics. But a bacteriophage is really specific, so it only infects the cells that it can specifically recognize. And in the literature so far, those are generally going to be pretty closely related to the bacterial pathogen. So it has this very narrow host range, generally. They're also incredibly numerous. So I said 10 times more numerous than bacteria, but what that means is this number like nonillion. So that's an incredibly large number that's hard to imagine. There are a lot of bacteriophages, and they are just full. The soil around us is full of these potentially useful viruses. And they're also recyclable. So once a bacteriophage is being administered, it is eventually going to get kind of turned over in the environment. So antibiotics, if we take an antibiotic, probably the estimates are that 80% of them end up in going through our bodies and ending up in the groundwater and in the soil, whereas bacteriophages are going to be turned over. But these can also be disadvantages, right? So that they're very specific means that we need to find exactly the right phages for the pathogen that we're interested in. And that can be a challenge, especially when you're up against a medical condition, for example, and you need that tailored bacteriophage. They're highly numerous, but we only know in terms of their DNA about 3,000 of these. So these have been discovered and sequenced and studied. So we don't know as many bacteriophages, like we've barely scratched the surface. And because they're recyclable, they're eventually destroyed. So if you are using bacteriophages in, for example, agriculture, you might need to reapply them. They will not stick around forever. So if we think that bacteriophages are something that can be useful, then we need to find the bacteriophages that will kill the pathogens that we're interested in destroying. And that's why there are like these assassins or these ninjas beneath our feet. So we got interested about 3 years ago in trying to investigate the degree to which we could solve the problem of American foul brood using bacteriophages. And of course, American foul brood is a disease of honeybee larvae that's caused by a bacterium called Penibacillus larvae. And you're all familiar with the symptoms of Penibacillus larvae and American foul brood. So there's the classic roping out and the larval tongue and coffee colored larvae. These are all signs that you might have American foul brood. This is what the microorganism actually looks like in our laboratory. And of course, the spores are one of the big problems in American foul brood, in that the spores have been documented to last for over 30 years in the soil. So we can grow the Penibacillus larvae now in our laboratory, and we can also turn the Penibacillus larvae into spores and back. And that's important for the kind of work that we're doing. So the reason that we think that AFB is something that can be handled by bacteriophages is that in 2017, there was a report in the Journal of Invertebrate Pathology. And actually this was, I'm going to just briefly describe one experiment, but there was a lot of really compelling evidence that if you put the bacteriophages that can infect Penibacillus larvae on a hive, that they can protect against that hive becoming infected. And so there was a natural experiment that was conducted where a single hive in an apiary of 11 hives was infected with AFB. And of course, in the United States, you're allowed to treat. This was taking place in the United States. So they were considering treating with antibiotics, and they decided instead to try treating half of those hives that had not been infected with AFB with these bacteriophages, and leaving the other half of the hives without bacteriophages. So they just subjected them to a sugar treatment. And what they saw 4 weeks later was that the hives that had not been infected with, had not been treated with the bacteriophages, most of those hives now had the AFB infection and were not protected. But the bacteriophages appeared to be guarding against the AFB spreading to those hives. And if they did the AFB Hitchcock scores, they saw that the mock treatment where they had received the sugar water, they had these high degrees of infection severity, whereas phage-treated hives didn't have any infection at all. So this is really a good sign that bacteriophages can be used to protect hives from getting AFB. So based on our interest in the apiculture industry and the evidence coming from the United States that this is a reasonable solution to AFB, we started the ABATE project, and that stands for Active Bacteriophages for AFB Eradication. And I recruited 2 students, Danielle Koch, who did most of the work that I'll be talking about today. She's been isolating the AFB causing organism, Penibacillus larvae, and discovering bacteriophages. And today I'll talk about the most recent work that she's done kind of in the last year, getting the sequences of these bacteriophages and testing the phage cocktails. The other student is Joe Turnbull, who was an excellent master's student, who has been developing an alternative host and also working on evolving phages. So the first problem, of course, for the 3 of us is that we're not beekeepers. So we don't have access to beehives, and maybe some of you participated in this project. So we have some beehives pictured here that are on the Massey University campus, but we started receiving soil samples and bee debris samples from beekeepers, but we weren't finding phages in the first instance. So Danielle, it's kind of it's almost a joke in bacteriophage hunting that everybody has to go to the wastewater treatment plant at some point and take some samples and try to find phages in wastewater. We didn't end up finding or even managing to process those samples because of our biosecurity issues. But what we did do in order to get access to samples was we asked you. So we received over 400 samples from across New Zealand of soil and bee debris from near hives. And we did that using this infographic that kind of made this call for, please send us these soil samples. And we had the cooperation of some of the other MPI-driven projects. And of course, we're working with AFB PMP, and we're working with the monitors of AFB. So from these 400 samples, we ended up finding 30 unique bacteriophages. And in order to do that, we brought soil or bee debris into the laboratory and filtered it for everything but the very smallest particles. And we plated the Penibacillus larvae on bacterial agar plates like this, and filtered those particles, so the very smallest particles came through and looked for evidence of bacteriophages. And these clearings in this lawn of bacteria are called plaques. And each one of these is evidence of a single bacteriophage landing on the plate, starting to replicate inside those bacterial cells, and then continuing to kind of amplify and amplify and amplify. And we have this exponential growth of bacteriophages in that little spot that eventually makes this clearing. So this is how we find the bacteriophages. And this is Danielle looking at plaques on a plate. And these are some of the phages that we discovered. So every time that a beekeeper helped us to find a bacteriophage, and we found a positive sample in one of their soil samples, we found a phage. They had the opportunity to name that phage. So phage Ali and AJG77 are both examples of names that have come from beekeepers. And I'll show you the complete list of names, but this is what the bacteriophages look like. So you can see that they have this, what we call prolate head and a long flexible tail. And this is, these are classified as Cyphoviridae. So that's the type of bacteriophage that these are. And that's the kind of distinctive look that we have to most of these bacteriophages that we found in this project. And we're still getting more images of some of the other bacteriophages, but this is just a representative sample. So that's a little bit about phage discovery, but there's a lot of work that has to happen after phage discovery in order to demonstrate both that the bacteriophages are effective and to understand some of their biology, and then eventually to get to this later stage of the project, which is testing the bacteriophages in actual larvae. So I'm going to talk a little bit about these two different streams of work that we've done. And I'll start with the host range, because you really need to establish the host range on the range of bacterial pathogens in order to start figuring out which bacteriophages you might want to include in a cocktail. All right, so this is an example of the 30 bacteriophages that we found in the project, and they have been color-coded. These are color-coded to the part of New Zealand that they're from. So we can see, you know, we're here in Auckland. So these kinds of light peachy bacteriophages were all discovered here in Auckland. And we named some of these bacteriophages when we didn't get names from beekeepers. But some of my favorite names, I think, are probably Bloomfield and Stretch. And we named a bacteriophage after Barry Foster. And we have also a Jacinda, who is from the Lower South Island. So this is an example of the host range of the full 5 bacteriophage, 5 bacterial isolates that we have from the North Island, and all of our bacteriophages. And what you can see here is that each gray square is where the bacteriophages are killing that bacterial strain. So these are 3 strains that are killed by any of the bacteriophages that we've discovered. And that's great, because that means that if that particular strain of AFB showed up in a hive, any bacteriophage that we exposed it to would kill it. Slightly more difficult are these 2 strains here on the right, 2006 and the 7957. These are only killed by Callan, Dash, and Lilo. And we do these host range tests by looking for plaques on plates. All right, so that's the North Island. We only have 5 bacterial isolates from the North Island at present. We have far more isolates from the South Island. And I have to mention the AP Wellbeing Project, but from MPI, has been incredibly important to us in allowing us to get our hands on 22 more bacterial isolates than we had previously. So Danielle had found 8 different bacterial strains in infected hive material. And these bacteriophages, I should also mention, have all come from healthy hives. So that's an indication that the presence of bacteriophages is supporting hive health. All right, so we have lots of strains from the South Island, and we can see that we have more strains that are only infected by Callan, Dash, and Lilo. We have lots of phages, lots of strains that are infected by many of the phages, especially these that are listed here at the top. These are great phages. They kill a lot of these bacterial strains. And what we discovered was that in the Lower South Island, there were 2 bacterial strains for which we have not found any bacteriophages yet. Yes, so all of these bacterial isolates that are listed along the top we don't know that much about all of them, but they're all Penibacillus larvae variants that have come from different samples of infected hives around New Zealand. So this gives us a little bit of a sense of how variable the Penibacillus larvae pathogen is in New Zealand. And what we want is ideally to put together a mixture of bacteriophages that would be able to kill one of these strains, no matter what strain showed up in your hive. And so one problem that we have right now is that we have 2 isolates, both from the lower South Island, for which we don't have any bacteriophages yet. And that's something that we're going to want to fix in the future. All right, so to summarize what we've learned about the host range, so far we have 33, we thought 33 Penibacillus, but it turns out that a couple of those were the same once we sequenced them. So we really have 30, I should correct that. And each one of these 30 bacteriophages is able to destroy between 53% and 87% of the Penibacillus larvae isolates that we see in New Zealand. We can see that we have this uneven distribution of Penibacillus strains, so we should probably get some more strains from the North Island, and we're in the process of doing that. And we've also found these 2 strains that are not able to be infected by any of the bacteriophages that we've discovered to date. Okay, so that takes us here. And we really wanted to do some DNA sequencing. And part of the reason for that is to understand how different these bacteriophages are from one another. Part of the reason for that is to see if we can see what types of infection they do. And part of the reason for that is that bacteriophages can sometimes conduct a life cycle where they contribute genetically to the bacterial strains. And so what that means is that sorry, I'm reading the question. So what that means is that sometimes we have genes inside the bacteriophages that are going to change some of the biology of the bacterial strains. And we might want to avoid some of those. So that's part of the reason that we do this genome sequencing. So I'm going to talk about this stream of work first, and then I'll talk about this lower stream. So we wanted to do the DNA sequencing. And so I have a question: why is it you can treat AFB in some countries but not others? And I think that the answer to the question might be the law. So in New Zealand, we're not permitted to treat AFB with antibiotics because the industry has decided that we don't want to encourage antibiotic resistance, and we don't want to introduce those types of chemicals into our hives and into our honey. But in the United States, you're allowed to treat with antibiotics. Okay, so in order to get the DNA out of our bacteriophages and send them to a genome sequencer and get the DNA sequence out, we have to have a high titer or a high concentration of those bacteriophages. And in this project, we were finding we could discover these bacteriophages, but they were in low concentration. So we could do the things that we would normally do with bacteriophages, and they were maybe 1,000 per milliliter. And what we really want is like 1 billion per milliliter. So we want much higher concentrations, both to use them practically and also to isolate their DNA and study it. So we needed to solve this problem. And we found this really interesting paper that was published in 2019 that talked about something that was called the modified Appelman's protocol. And this is something that they have been using in Poland to do like an experimental evolution of these bacteriophages to get them to swap bits of DNA between them. And that had the property of expanding the host range if you had the right bacteriophages. And so we looked at this and just thought, well, I mean, we could change this a little bit. And instead of mixing all of the bacteriophages after they've done lysis in a 96-well plate, we could just take this group of bacteriophages that grew in these different dilutions and put them back on the same bacterial host again, and just keep doing that again and again. And that might evolve the bacteriophages to be better at infecting that host. And if they're better at infecting that host, then maybe they'll be better at increasing their concentration. So the Appelman's protocol is normally done for about 30 days, but we saw that in as little as 4 days of doing this repeated infection in these 96-well plates, we could find huge increases in the concentration of bacteriophages. So we had, these are just some examples of some of the bacteriophages. They were maybe 1,000 per mil, maybe 10,000 per milliliter. And then after 4 days of evolution, we could get much higher concentrations, so millions per milliliter. So 10,000-fold improvement in the concentration in as little as 4 days. This for us was just unbelievable. So we were really happy to find this kind of improvement and such a simple protocol that just took, 4 days in the laboratory of propagation. So this was really a game changer for us. And it allowed us to extract the DNA and start sequencing these bacteriophages, which changes the game for us. So we managed to do some experimental evolution, which is going to lead to small nucleotide changes that occur randomly in the bacteriophages, and then select for the ones that are improving their ability to infect. So that was great. So next we wanted to take the information about host range and also what we were learning from DNA sequencing and design some bacteriophage cocktails. And again, the idea here is to find a set of bacteriophages, that would kill most of the bacterial strains that we might find in a hive. So Karen asks, the principle of survival of the fittest and perpetuating the strongest lines. That's exactly right. So we were allowing evolution to take place over the course of 4 days in these simple little 96 well plate liquid experiments. So yeah, that's every time anything replicates its DNA, it has chances of mutation. And what we were doing with the experimental evolution is just allowing the mutants that were a little bit better to win out in this experiment. And that's something that we do in my laboratory a lot. So it's kind of fun for me to bring these two halves of my interests, evolution and bacteriophages, into the same project to solve a problem that we were having. Okay, so let's talk a little bit about cocktail design. There are lots of ways you can imagine taking this set of bacteriophages and coming up with a small number of them that you would put into a mixture, and then that would be the thing that you would want to put on hives to protect them. And what we eventually ended up doing was deciding that if we had any of these strains of Penibacillus larvae, any of these bacteriophages would do. But if we choose the ones kind of higher up in this list, they also have the chance of killing possibly these green strains. Some of them, if you picked them, would also kill the red strains, and some of them, if you killed them, would also kill the purple strains. So we've listed them in this order so that they have these qualities. But we thought, well, we'll make sure we have something from this blue group and something from this green group, something red and something purple. And of course, in the future, we'd also like to add something that would kill these outliers that we don't have any phages for at the moment. So and then there was one more piece of information. So the project that Joe, the master student, had been working on allowed us to see the genome for Lilo before we could see the genome for anything else. And of course, in this purple group, this whole set of strains, including strains from the South Island and the North Island, have to have either Callan, Dash, or Lilo in the mix in order for those to be infected, for those strains to be infected. So that gave us some important information, and that was that Lilo is probably not going to be a good choice for our bacteriophage cocktails. So we came up with 4 different cocktails to try. Callan, Logan, Matsy, and Freya were in cocktail 1, and you could see the names of the other ones, and you could see these kinds of color codes that describe which sets of strains they can infect. And we wanted to try all 4 of these against some small number of bacterial strains that are the examples of what you might find in a hive. So the way that we did this was, again, to use 96 well plates and to just set up a serial dilution of the bacterial strain of interest. So here are the bacteria, and also serial dilutions of the bacteriophages. And we had, all of these wells had either the bacteria or the bacteriophages, or both. And so in this area, you can see the results of having both. And then we just, after we did the infection, we let that go for 24 hours or 48 hours. And then we took a tiny amount of that liquid, and the white bits on this plate represent single cells that had survived that infection. And in this red box, these are the numbers based on the literature that you would see probably of bacteriophages, around 10 to the 5 you could get into a hive. And this is the dilution of the bacteria that you would generally see in a hive. So that's the box that we really kind of want to use as the guide to see whether or not this is working. We want to see as much cell death or as few bacteria growing here as possible. So this is an example of 2017. And here we're infecting with cocktail 1. And if we expand that, we can see cocktail 2 and 3 and 4. And what should be fairly obvious here is that cocktail 2 and 4 appear to be performing a bit better than cocktail 1 and 3 in terms of clearing the bacteria on this plate. And cocktail 2 and cocktail 4 seem to be doing really well. Ooh, Karen is asking me about viral reassortment, and that's something that we would like to look into more, using that Appelman's protocol. So using that experimental evolution approach, we hope to improve some of these phages further by doing recombination in the future. But that's a great question, and one that's very close to my heart. So we tried these different cocktails out on 4 different strains that were kind of representative. So here's the 2006 strain that we find here in the North Island. And you can see again that cocktail 2 and cocktail 4, with a little bit of contamination, it looks like in our experiment, are really doing an excellent job of killing this Penibacillus 2006 strain. So again, cocktail 2 and cocktail 4 perform better than cocktails 1 and 3. And then 8100 is a South Island strain. And again, in the area that really matters, we're seeing pretty good results with cocktails 2 and 4. And of course, P1627 is one of those 2 strains from the South Island for which we don't have any bacteriophages. And we wanted to try this just to see, like, if we put these bacteriophages in combination on this strain and we don't think that any of them are going to be able to infect it, do we see any kind of emergent property of this mixture suddenly doing a pretty good job of killing? There are some biology reasons that that kind of thing might happen, but actually we don't see that in this case. So it really looks like we're going to have to find these bacteriophages to kill P1627 and the other South Island strain. So about at this point in doing our cocktail testing, we got back some of the genome sequences for the first 6 bacteriophages that we had put through that Appelman's protocol and then annotated. And so what you can see here is this set of 6. It includes Lilo and Dash and Callan, Abate and Zed, AJG77 and Loken. And these purple bands between each of the phage genomes represent areas of high similarity. So Logan and AJG77 are incredibly similar to one another, but a little bit different, and so on and so forth. And what we were kind of devastated to see is that our phage cocktail that was doing so well, phage cocktail 2 and phage cocktail 4, both have Dash. And unfortunately, Dash has this same honeybee toxin, this larvin, that kills honeybee larvae that Lilo has. And so we wanted to see if, in order to avoid putting together a phage cocktail that has this honeybee toxin in it, and increasing the numbers of copies of that gene in the environment, if we could replace the Dash phage with the Callan phage in cocktails 2 and 4. And unfortunately, as soon as we replaced and took Callan out, as soon as we put Callan in and replaced Dash so that we wouldn't be adding more of this larvin, we saw much poorer results. And we're not exactly sure why that is right now, but to Karen's question, we would love to do some more recombination experiments with these phages and see if we can get the balance of qualities right, where we have Callan, which doesn't have the phage, the larvin, this honeybee toxin in it, but does have the qualities that make it so good at killing the bacteria in cocktails 2 and 4. So using these types of experimental evolution approaches are ways that we can hopefully get the right combination of traits by doing recombination with these bacteriophages. So that's a great question. All right, so we have sequenced 8 of the NZ bacterial isolates. And I know that the AP Wellbeing Project is in the process of sequencing the rest. And I can say that all of those Penibacillus strains have this larvin toxin in their genomes, but I still don't like the idea of making a phage cocktail that has a toxin in it and exposing honeybees to something like that. So this is an ongoing issue for the project that we are hoping to resolve, and we would like to do more work on this in order to figure out if there's a way that we can get the balance right in the traits. So the 2 work streams that I've talked about were the experimental evolution and the DNA sequencing. I hope now that you can see why the DNA sequencing is so critical, so that you know what the traits are of the viruses that you're intending to use to protect a hive. We've gone through some cocktail design and some in vitro cocktail testing where we tested them in 96 well plates, but you never really know from a 96 well plate experiment what it's actually going to be like for a honeybee larvae to be exposed to these bacteriophages. So we have some wonderful colleagues, Ashley Mortensen and her team over at Plant and Food Research. And this summer, since now kind of spring is passing us by and we're still in lockdown here in Auckland, we're going to be sending Danielle Koch to the Plant and Food Research facility, and she's going to be rearing honeybee larvae in 48 well plastic plates. And that's going to give us the opportunity to expose them to the bacterial pathogen, and to, in advance of exposing them to the bacterial pathogen, to give them the bacteriophage cocktails and see how well do the larvae respond to the presence of the bacteriophages alone, the bacteriophages, and then the AFB pathogen, or the AFB pathogen alone. So we're really excited about doing those experiments because we think it's really going to help us to understand what the bacteriophage cocktails will be like in a live hive. And of course, we're hoping that experiment will happen in January now that we're hopefully not going to be in lockdown forever. So that's still to come. Okay, so the summary of the latter half of the talk is that we've identified some effective cocktails for most New Zealand strains of Penibacillus larvae. So we think that we are pretty close to being at the point where we could say, here's a bacteriophage cocktail. If you put this on your hive will be healthier and safer than it would have been otherwise. Of course, the conundrum right now is what to do about this toxin, and can we eliminate this toxin from these bacteriophages and come up with the perfect mixture of bacteriophages. So moving forward, we are going to be testing these cocktails and trying to eliminate the toxins that we find in some of them. We'd like to isolate more bacterial strains from the North Island, and we'd also like to find bacteriophages that will be able to infect the 2 strains from the South Island for which we don't have any bacteriophages yet. And ultimately, we are going to be partnering with groups like Comvita and some of the apiculture training courses to perform blind field trials, where we will either apply a phage cocktail or a sugar solution and look at how well the bacteriophages protect those hives against AFB. And of course, I'm in the process of finding more funding to continue the project, but we have been funded by both AgMart for the next phase of the project for the next 1.5 years, and also the Honey Trust. And we're incredibly grateful for both of those sources of recent funding. All right, so in wrapping up, and then I can take some more questions, I really think that bacteriophages are incredibly promising for solving problems in agriculture. And I would love it if 10 years from now, we're not only solving problems in agriculture with bacteriophages, but we have grown our intellectual and physical infrastructure to the point where we might be able to solve medical problems with bacteriophages. As we see more antibiotic resistance evolving in bacterial pathogens around the world, I think that this is a really promising source of natural solutions to the problem of bacterial pathogens. And so with that, I have to say, you know, I've been at Massey University for 10 years and it's been great. I am now moving to the University of Canterbury in basically in a month. So by January, me and my laboratory will be at the University of Canterbury, but we will still be thinking of our friends here in Auckland and working to find solutions for your beehives as well. And I have to say, you know, AgMart, the Sustainable Farming Fund, have contributed to this project in the past, and the Honey Trust and also AgMart, again, are contributing to our future for this project. We're grateful to HHMI for the training and for the science education platform that they've allowed us to be a part of at Massey University. And while I love bacteriophages and I am excited about this work, we also do other types of projects that involve bacterial evolution, and that work has been funded by the Marsden Fund. And with that, I should be able to take some of these many questions. So I think I will stop sharing for the time being, and let's have a chat. Thanks.
There are several questions there on the list. Do you want to simply pick up on those and answer them, Heather? Are you comfortable going through those?
Yes, it should be good. Hopefully I understand them all. And if I don't, I'll have to get some additional comments. So Peter says, can you say more about larvae? What is the advantage for a phage carrying the larvae gene? So one of the things that I'm not talking a lot about right now is the fact that bacteriophages also sometimes have another life cycle where they integrate into the bacterial chromosome and just hang out for a while. And you can imagine that, especially in a situation where there aren't a lot of hosts, if there aren't a lot of bacteria to infect, just sitting and replicating along with your bacterial strain can be a really successful way of living. So bacteriophages will shift between these two life cycles with some frequency. And so for a bacteriophage, if it has a larvae gene in it, that might make the bacteria that it sits in slightly more pathogenic, and that might be good for the bacteriophage once it's integrated into the bacterial chromosome. So that's why they have the larvins. Bacteriophages, more than anything else, are a little selfish genetic element that carry with them tools that make them better at replicating. And so that's the story on the larvae. And but we can propagate bacteriophages that don't have the larvae because we're the ones that are choosing what traits they have or don't have. It's going to be really problematic for us to get into genetically modifying bacteriophages for a lot of reasons, which is why we really like the idea of using these recombination experiments and trying to tip the scales of natural selection towards choosing bacteriophages that don't have the larvae. And one way that we might be able to do that in this case is by choosing bacteriophages that have less DNA. So they might be able to replicate faster or in higher numbers if they don't carry that larvae in this kind of artificial setting. So those are the types of tools and tricks that we'll be using in the future to try to get away from bacteriophages that have larvae genes. Okay, so Karen asks, are the cocktail solutions applied as an aerosol to cover surfaces or feed the bees to ingest and spread? So this is a great question. Thanks, Karen. There are lots of ways that you can imagine administering bacteriophages. In some instances, and we haven't started to get into that side of the project. In fact, I'm applying for some IMBY funding, hoping that we can start investigating how do you apply the bacteriophages that we have? How stable are they? How long do they hang around? Those kinds of questions. So you could put them in a liquid solution. So you could put them in like a water trough nearby, or you could potentially do something called lyophilization, where you make them basically into a dust and then you can dust. The key is going in whatever administration technique you use, and some people have used sugar water, but I can see why people might want to steer clear from sugar water if you're really trying to get honey that has these native or non-native plant characteristics. So there are lots of methods that we can use, but the trick is going to be you've got to get the nurse bees administering the bacteriophages to the larvae. And so that's really the critical point in making sure that those bacteriophages are in the place that they need to be in order to protect the health of the larvae. So there are lots of ways that we will be trying that. I know people have tried smoking the bacteriophages into hives or tried water solutions and sugar. Okay, another question from Karen. How do the phages survive if there are no host bacterial targets available? Yeah, so with these bacteriophages, we don't know right now if they are able to infect anything else in the environment, and that's something that we could investigate further in the future. And that's part of the reason that if the bacteriophages are present and the pathogen shows up, then they can amplify on it, and then we'll have more of them. But that is exactly why we need to test how long do the bacteriophages hang around if there isn't any AFB present in the hive. And that's something that that the INBI project that we're trying to get funded would also investigate that question. So that's something that we would like to know as well. Is the toxin a byproduct of the bacteriophage life process, or is the bacteriophage DNA directly toxic to the larvae? So a bacteriophage, as it's kind of floating around as a little protein particle, doesn't have any energy, doesn't have any ability to replicate itself, doesn't have any ability to produce toxin or excrete anything. Like it is actually a matter of debate. Here, I'll got my little hot pink bacteriophage here on my desk. It's a matter of debate as to whether or not a bacteriophage is alive, right? Like, so it can't do anything until it infects a cell, and then it can only produce proteins and make copies of its DNA and do all the things it needs to do once it's using the energy of a cell. So, so a bacteriophage itself, if it has the larvae in its genome, it's not doing anything. So it's not, the larvae isn't a problem until that bacteriophage integrates into the bacterial chromosome and is sitting there and is being expressed by the bacterial cell itself. And that's when the larvae becomes a problem. And what I would hate, because I've studied this process of horizontal gene transfer where bacteria, partly through bacteriophages, exchange genetic material. What I would hate is to see us use a bacteriophage that has a larvae in it, and that larvae start ending up in more bacterial strains than it's in right now, and increasing the number of things that are pathogenic towards honeybees. So that's why I think sequencing is so important, and really knowing as much as possible about the biology of these bacteriophages before we start using them is absolutely crucial. And that's a discussion I'm having with some of my feistier colleagues at the moment. So that's an ongoing battle. All right, Tim asks, does the presence of bacteriophages in the hive account for the antibiotic properties of honey? And if so, are these phages properties influenced by honey types such as Manuka? So there's a lot in that question. The antibiotic properties of honey is especially of Manuka honey, has been studied, and it's down to a chemical that I'm sure somebody in the chat remembers exactly the name of the chemical right offhand, but I don't. So it's a combination of the work of the honeybees and the Manuka plant itself to produce that little chemical, and that's what actually makes honey have antibiotic properties. So it has antibiotic properties against like Staphylococcus and E. coli, and especially in bandages and things like that. So it's not because of the bacteriophages. If so, these phages properties influenced by honey types such as Manuka. One question I would love to know, and I'd like to get together with some of my other colleagues that work on the microbiomes of floral plants, is, where are AFB in the environment? So where is the Penibacillus larvae hanging out in the environment so that it gets into the beehive in the first place? And it may be that It may be that the bacteriophages that fight against AFB are present in things like Manuka. It may be that the honeybee pathogens are hanging out in things like Manuka. We just don't know where the natural reservoirs of these pathogens are, and I would love to know that. But right now, I don't have any idea. So I don't know where the bacteriophages are hanging out, except that in New Zealand, every bacteriophage that we have isolated from hive material and from soil have only come from healthy hives. And we have sampled a lot of unhealthy hives, and we have not found any bacteriophages in them. So I think that that's really an interesting kind of factor. Certainly, I can say that in the United States, they did find some bacteriophages against AFB in AFB infected hives, but that's not been the case so far here in New Zealand. Okay, is the aim prevention or treatment in hives? Jackie, that's a very tricky question. So we are, of course, in New Zealand, not permitted to treat hives that are infected with AFB. Any hive that's infected with AFB has to be doused with petrol, and it has to be burned within 7 days of discovery. So because those are the regulations here in New Zealand, we would never suggest that anyone should try taking a bacteriophage cocktail to treat a hive because it's not legal. But what we can do is we can put these bacteriophages in the hives in order to protect them against getting AFB in the first place. And you know, in the natural experiment that they did in the United States, of course, they saw that if a single hive had been infected and you protected the other hives in the area, then you were less likely to see the spread of that AFB infection. And without getting into how AFB gets between different hives, I think that that's the idea that we could protect, especially if we think, you know, maybe a neighbor has AFB, or if we're in one of those hotspots that the AFB PMP has reported for the country, or if we're starting to get the idea from some of these reporter apps that we might be at risk, that would be a great time to start protecting with a bacteriophage cocktail. All right, Peter's got a question. Would it be possible to synthesize a minimal phage to bypass the genetic modification problem? I think most people, I mean, that's a good question, Peter. I think most people are going to consider, I mean, genetic modification in New Zealand has very, it's very loaded. And I think in an industry where we are known for this kind of pure natural product, if I did find some way of synthesizing the DNA and then getting a little tiny bacteriophage made that didn't have any negative properties, I think people in the industry would probably be very wary of that. But if I can say, I have taken these bacteriophages from healthy hives, I have naturally evolved them to not have any of the negative properties, and I have tested them, I think that that's something that, in my impression, is that more people will be able to get behind that kind of a natural protective product than something like a synthesized or genetically modified bacteriophage.
Thanks very much indeed, Heather. I think you're probably quite right. Adding genetic modification into what is supposed to be a healthy natural product is almost a contradiction in terms. To go back to, I think, one of the first questions about why we don't treat in New Zealand, I found a very early account of beekeeping in New Zealand, 1870s through to 1914, in the club's archives when I was doing a cleanup. And there are references in there to AFB having arrived in the country and being found, and actually having infected the main hives at Ruakura, the research centre and so forth. And of course, this occurring 1914 or prior to 1914 is well before antibiotics had been discovered. So I think simply the regulation came out at the time in New Zealand. There wasn't a way of treating with antibiotics against AFB, and therefore the regulation was introduced to say you burn the hive, and the Americans subsequently amended to say, oh yeah, we can treat with antibiotics, and New Zealand has simply maintained its previous standard of saying, no, we'll just destroy the hive, and that's the way we go with it. So I think that might have been one of the contributing factors to the way in which we approach it and how we're different to the rest of some of other countries. That's outstanding.
I think we're doing ourselves a favor. I think we're doing ourselves a favor by not using antibiotics, because that will naturally select for antibiotic resistance, and that's a whole heap of trouble.
Yeah, indeed. That was fascinating, both for what you're trying to do, the progress that you've made, but also for the stages that has to go through. I don't think any of us looking from outside, unless we have that sort of laboratory or research background, have an understanding of the difficulties that are involved in and the steps that you have to go through between having a good idea at the beginning and then coming up with a product at the far end. So I was fascinated to actually look and see what you're doing, how you're doing it, what you have to tackle, and all the different steps to it. Thank you very, very much indeed, on behalf of all of us sitting in and attending and listening. And we wish you the very best, both in your personal, your move down to Christchurch in Canterbury. It's a lovely city. Having been born there, but I'm biased. But also all the best for your work in the future, and our best wishes that you find a solution for a product that I know has, or a solution for a problem that I know has devastated many beekeepers over the years, who have worked hard in their hives through no fault of their own, have found to have AFB in their hives, and just the devastation of having to pour petrol through your hive, burn your girls that you've worked so hard to keep. If we can find a better solution for it, then you will have the eternal gratitude of both this club and beekeepers all across the country and elsewhere as well. Thank you very much indeed for today.
Thanks, guys. It was a real pleasure. And if you have more questions for me, you can find me online. And it's always a pleasure to hear from the beekeeping community. It's really been fun to learn more about all of you. And you had great questions. So thanks a lot.
Thanks, Heather. Enjoy the chocolates.
I'm really delighted. Thank you so much. Cheers.
Cheers.
Today I'll be talking about smoking and smokers in the context of beekeeping. The smoker is, after the hive tool, the next most important piece of kit for a beekeeper. We smoke them, the bees, to calm them down so we can go into the apiary without getting stung or without them getting too aggressive or upset. It's believed that this is an old reflex from after having spent tens of millions of years evolving in trees. The smell of smoke makes them think that there's a fire coming, so they will go onto the frames and they'll start gorging full of honey, ready to evacuate the hive. So for about 15-20 minutes after they first smell the smoke, they will just go straight down onto the frames and start gorging on the honey, so it will calm them down. Also, it interrupts their pheromones, because the bees, they communicate with pheromones in much the way, same way as we communicate with speech. So the smoke interrupts the pheromones, they can't communicate with each other, so they go to the instinct of gorging themselves on honey. So this is a typical smoker, quite a small one. There's three main parts to a smoker. There's the spout, the combustion chamber, and the bellows. The bellows' purpose is to pump air into the combustion chamber. And you will notice that it's not directly connected to the combustion chamber. So when you're not pumping, there's still a flow of air through the bottom, heat rising up through the top. So it'll keep it going. And the bellows will pump extra air into there to make it go more. And then in the fire chamber, we have a little disc, usually in here, which keeps an air gap underneath so that the air can go under the whole part of the chamber. And then you'll notice that it's surrounded by a cage. Almost every single smoker will be surrounded by a cage because that gets very, very hot and it will burn. So you need to be very careful, especially when you're trying to take the lid off if it's jammed, because sometimes unburnt tar and resin will go around the edge and make it very hard to lift off. So you have to be very careful about not burning yourself. And then there's a hinged lid with a spout, and that spout is where you direct the smoke when you are smoking the bees. And sometimes there'll be a hook on there, so you can hook it on the side of the hive so it's out the way. So the next most important part of the smoker is the fuel. Now it's always a toss-up between burning fast and burning for a long time. So you want it something that will burn, but then something that will smoulder. So it's quite a good idea to have two separate things. Like I tend to go for paper and sacking. So the paper gets it going and the sacking keeps it burning. There's two types of sack. We use coffee sacks. They tend to be the best one, and they're available from coffee shops that roast their own coffee, and on Trade Me or even Hive World. Some of the beekeeping supplies supply them. They're usually $1 or $2 each, and a sack will last quite a while. There's two types of sacking. There's Hessian, which is a light, easy to burn sack. And then there's jute, which is a much coarser, thicker sack, which is a lot harder to burn. The best way to prepare it is to wash it, just in case there's any chemicals. I once put it through the washing machine, which was a big mistake because all of the fibres break off and it makes a mess in the washing machine. So it's not a good idea to put it through the washing machine. If you do, then put it in a bag that you put a dog blanket or a horse blanket in to keep the fibres in there. I've discovered the best way to do it is to put it in the bath and put some detergent in there and put the plug in and fill it up with water, and then you can just stamp on it like you would be stamping grapes when you're making your wine, and then you just pull the plug out, let it drain out, and then fill it up with clean water and repeat the process, and then you'll end up with nice clean sacks. One thing you can do is to weather them, the sacks. You just leave them out over winter so that they will rot, and then it makes them very easy to use. And some people only use those, but you can tear them, and you know when you can tear them quite easily, then you know that they are ready to go. So also some people use cardboard. So a piece of cardboard and rolled up corrugated cardboard. If you roll up and then put that in the chamber, that will burn slowly. With the sacking, I use organic material as well. So cut grass is good. It burns very quickly, but it gives a very good, nice smelling smoke. Pea straw is very good. The only problem with pea straw is you can get the bits sticking out when you're trying to close the lid, and that becomes a bit of a pain. And a great thing is pine needles. They smoulder really well and give a nice smell. So back to the very end, when you finish with your smoker, there's still burning material in there. So you might just be putting the smoker in the shed, or you might be putting it in the car to take it away. So some people will open it up and empty it into a tin, like an old coffee tin, and put the lid on so it'll go out. Or some people just put it down to one side and let it go out. I find putting a piece of grass in the end, or a cork, just cut down a cork slightly. You might have to buy a slightly nicer wine instead of the screw top, but bit of cork in there, and that stops the combustion and it goes out. And then next time you go to do your smoker, you're left with the equivalent of charcoal, sacking charcoal, and that burns as good as paper. So it's a very good way of starting your fire next time. So what we do is we just put a bit of newspaper, and I find that this way of lighting the smoker, it will last me all day. One match and I will not need to relight it again. I need to keep adding to it, but this is a foolproof way for me. So just one match, as long as it's not too windy. So let it burn, maybe put another piece of paper in. Pop the bellows to get it, there's a bit of ash still in there, but so the fire's going. Then we put in some sacking and get the fire going. If you're going in for a short time, don't be fooled by just getting it going a little bit. It's got to be going properly no matter how much you're using, otherwise it will just go out and then it's no good. So now you've got the flames in there, so the flames are coming out. So put some bit more sacking in and you can push it down a bit, and then you put your organic material in. So I'll start off with a bit of grass cut and then a bit of pine needles, and then just pump it to make sure it's going nicely. So you know that once it's going like this, you know that it's going properly and it's not going to go out. And then the trick is to put a piece of sacking on top. Now the point of this is that it will help cool the smoke that comes through and stop any sparks coming out, because you want to be careful about it being too hot or sparks coming out, because you could burn the bees' wings, and then that's a death sentence for the bees. So and just pump it to get it going really, really well. And once it's going like that, you put your hand on it and test it. You can, if you've got your glove on, just test it on your face. But you just make sure that it's going well. And if it's flames still coming out, it's hot, just turn it upside down and back that way, and that puts the flames out. And then it's going good, and then you can leave it and it'll be fine. And then it'll slowly burn down, and then what you can do is you just, once it's burnt down a bit more, you just put a bit more sacking on. Half an hour, 3/4 an hour later, and then you just do that through the day. Keep going like that if you need it for the whole day. But the crucial thing about a smoker is to make sure that it's going properly first, because if it's not going properly first, then it's just going to go out. And when you're doing your hives and you're checking them, the worst thing is if they start getting angry. Maybe you've squashed a bee and it's put out an alarm pheromones, they're starting to get a bit angry, and you reach your smoker and it's gone out. And then, so you've got to then try and spend 10 minutes lighting your smoker, getting it ready. By the time you go back, you've got an angry hive. But if your smoker's always lit, if you're not using it for a while, say they're calm, that's fine. Just every now and again, just check it, give it a squirt, just to make sure that it's still going. With regards to fuel, don't use anything toxic to bees. Don't use plastics. Don't use any accelerants to start it. No petrol, meths or terps or anything like that. Just use paper and nothing that's going to hurt the bees. Now also you need to be careful. I've mentioned about the risk of fire and burns. That gets very hot. So if this is sticking, you're trying to open it up, you can just use the hook of your hive tool to lever it up and to open it. Sometimes it does get a bit sticky, so you can just pull it up like that. And sometimes if it gets up there, what you can do is you can just scrape it around, and then that helps you next time. If it's windy, you need to light it in there instead of using a match. One of these lighters is quite good because you can light it inside there. Another thing that's quite handy is one of these. So a blowtorch, that will get it going pretty well, but don't fall into the mistake of putting everything in there and then lighting it with this, because then it will be burning from the top, and it will be harder when you're blowing the air. The heat's going to be at the top, not the bottom. It's not going to be burning through. And another good thing about the blowtorch is that you can use it, because you do get a lot of build-up inside, so you can use this to burn all around there, and then use your hive tool to scrape all the unburnt stuff off. And this also is great for sterilising your hive tool if you're going to another hive, rather than scrubbing it with soapy water, and that will kill any risk of AFB. But once again, be careful with this as well. So another thing that I find useful is with my toolbox, I've got a separate compartment for the smoker. That way, I put it in my car, put the cork in, put it in my car, and it's not going to fall over and it's safe. Sometimes it might be going just too much and it's just going crazy and you want it to slow down a bit. Putting on its side slows it down a little bit, or you could just put your cork in for a little while just to slow it down. But then you think, oh no, I've just gone through another piece of sack, and that's like, you know, the third piece of sack and, you know, doing this hive. But then the sack only costs $1 or $2. It's not, you know, it's more important to make sure that this is ready and prepared and ready to go the whole time. Now, sometimes also there's the risk of fire with sparks coming out, or say a careless beekeeper might just empty out the ashes and then start afresh. And that can start a scrub fire, bushfire. Maybe it won't affect you so much if you're in suburbia, but it's always a good idea to have a hose or a bucket of water just in case, even just to put your hand in if you get burnt, but also just to throw it on if it catches fire. If there's actually a fire ban and you can't use a smoker, then an alternative is water. You can spray the water on the bees and it will slow them down. I find that when I'm they can start getting angry and they're in the air, you spray smoke around, but the wind blows it away and then there's nothing, they're not calming down. But I find that spraying water on them around you if they're getting a bit upset will slow them down quite a lot. So some people do that. Also, what some people do is put a light sugar syrup in here. So, and spray it on the bees, and then they will stop and clean the sugar off themselves and clean it off each other. So that'll calm them down a lot. It's a good idea in spring and summer, but be careful in autumn because then it's a much bigger risk of robbing. So when you come to your hive, what I first do when I'm going to open it up is I just knock on the door. I just put a little spray of smoke in the entrance. If there's a mesh base, I just go underneath and give it like that. And then I'll take the lid off and then I'll just crack open the hive mat just a bit, put the spout in and just give it another puff, less than that, little puff, and then put the hive mat down. And then just give it 10 seconds, 30 seconds for the smoke to permeate through the hive and just for them to know fire's on its way, time to start gorging on the honey. And then when you lift it off, they're a lot calmer. And it's quite good practice to take off the supers and start at the bottom box, because if you're smoking them, a lot of them will go down to avoid the smoke. And then if you're checking the first box and then you take that off, you check the second box, and each time you're driving half the bees down. So by the time you get to the bottom box, the brood box, where that's the most important thing, where you want to check the bees, check the queen, check the brood, there's loads of bees there, and by this time they're getting a bit annoyed, and so it's a lot harder. But if you take each one off first and start at the bottom, then it's a lot easier, and you just give it a little smoke. I give a little smoke around the outside, because if you smoke in the middle, give it a good puff, it'll drive them outwards and onto the sides, so then there's more chance of them getting squashed and it's harder to see the queen. So just a little bit round, and then when you've done that, put the next one on and work your way back up that way. So just remember, with beekeeping, preparation is essential. So if you have a well-maintained smoker and plenty of fuel, then that's one less thing to worry about, and it will make your beekeeping much more enjoyable.
Thanks, Ken. That's a good one. One, since it's coming up and it's the appropriate time of year, Ken was mentioning about the pine needles. I use them a lot, and I get them from Christmas trees. So grab a Christmas tree, either from yourself or friends, neighbours, family, whatever. Lay it out in the sun for a week or two, let it dry off. You'll find that once it's dry, the needles come off the branches very, very easily, and then you can put them together into a box. And I find 1 or 2 Christmas trees gives me enough pine needles for years of smoker filling. So that's just one I found works well. Righty-ho, thanks to Ken for that one. And what I'm going to do is pick up now, and we're looking at supering up. So looking at managing the hive over the nectar flow, and also while you're away on holiday. First thing I want to say is be aware of the differences that will occur with the timings for the various activities here based on where you are in Auckland. So if you're in suburban Auckland, the main sort of metropolitan suburban area, then we've had nectar flow going for the past couple of months now. A little bit patchy when there's a rough day, as we're having today outside. It's still raining again here, but it's been running for a couple of months now. If you're further out into the rural areas, say going up Taupaki or south, or if you're up higher, going into the Waitakaris, where you're several hundred metres above the main sort of Auckland area, then your nectar flow is much later. And I was inspecting hives out in Waitakaris and Taupaki, what would it be, say about 3-4 weeks ago, and they were still sitting quite happily on a single full depth box and weren't looking to swarm or burst their banks or do anything else like that. So the actions that you require are basically, the principles are exactly the same no matter where you are, but the timings are going to be different depending on where in Auckland you are. So be aware of the difference that either elevation or a rural environment makes to supering up and how quickly your hive will build up and when the nectar flow occurs. So I want you to look at the hive as being an entire space. And the first question to look at here is, are you going to use a queen excluder or not? If you are using it, then make sure that you're providing sufficient space for both your brood area and your honey supers. And this is critical because you're obviously wanting sufficient good brood area so that the queen is not constrained in her laying and she can build up on the back of a good nectar flow, warm days, drones and everything else like that. You want sufficient room there so that she doesn't feel pressured to swarm. And you've also got to provide sufficient room also for your supering. I have seen 2 hives recently when I've been doing inspections for AFB, for the COI, where there was insufficient room within the supering, and the bees were therefore starting to backfill nectar into the brood area. So what had previously, and we know the traditional sort of shape of the brood frame, a central area of brood and then pollen and nectar around the outsides. But what had happened was the bees had emerged after their 21 days from that central brood area. The beekeepers had not provided sufficient space in the super. The bees had then backfilled into that central area with nectar, simply needing to put it somewhere. And the queen was then forced to laying on the very outskirts of these brood frames. And in one of the hives that I'd inspected, there was actually the beginning of swarming with queen cells appearing. In this case, the beekeeper looking at it and seeing patchy brood around the outside of the brood frames and cells, queen cells appearing, misinterpreted and thought that in actual fact this was supersedure and was about to replace the queen with a new one, believing that the old queen was in fact end of life and here the hive was going to replace her. So all we had to do there was say, put another super box on, provide sufficient room up in your supering area for your nectar, and the bees will move it from the brood space and up and into the super, and you've just saved yourself $50 and the whole time and hassle of replacing a queen. So make sure that you're not just putting on extra supers, but that you're also keeping an eye and making sure that your brood space has sufficient room in it for the queen to be active and to laying as much as she wants to. It's one of the things about a queen excluder. Some nectar will be placed below the queen excluder because, as we sometimes refer to it, it's a honey bar or a honey sort of barrier. So some bees, some of the workers and the house bees will want to actually put the nectar down below. So just make sure that sometimes when you're keeping an eye on your supers, that you're also looking down into the brood area and just making sure that they're not putting too much honey down there and there's sufficient space there for the queen to lay. So if yes, providing a queen excluder, space for both brood and honey. If no, the bees will tend to chimney the brood up through the middle of the hive. And that may not be an issue for you. You simply have to separate brood from honey frames when it comes time for extraction. Generally, we're looking a full depth or a 3/4 for brood. We were trying an experiment a year or so ago with putting half depth boxes on for brood, but queen didn't really like it, so don't recommend that. And looking at a 3/4 or a half depth for supers, a 3/4 depth box will be about 23 kilo when fully filled up with honey and capped. If you're putting a full depth box up there, you're up to about 30 kilo. And if that's up sort of your 4th or 5th box up on the hive and up at shoulder level, then trying to lift, unstick, unpropelize and lift off a 30 kilo box at that height is not a good idea. So generally a 3/4 or a half depth for super. Supers can be as many as your bees and your location can fill, and it also depends on your extraction plans. Whether you're doing a progressive extraction, so you're allowing the bees to fill a box up, and when you've got a good box of filled capped honey, take it off and do the extraction, or whether you're doing your extraction in a big session at the very end of the season. So that determines the number of supers that you're wanting to use there. Space in the supers. Don't wait until your super is full before adding another. If you're doing that, you've left it too late, because we're remembering, of course, that the bees not only are storing the honey, but they're also removing moisture from the honey. And so they want to spread that honey out a bit, dehydrate it, lower the moisture content, and that therefore and they will move it around, add enzymes to it and so forth as they go. And so you're actually looking to probably add another box when your previous box is about 50% full. So that's the 3rd item down. Add another super when the current is about 50% full. Remembering to rotate your frame, center to outer and outer to center. As I say, the bees tend to chimney up through the middle, and your super might have 4 frames fully capped in the middle of the box. And you get to the outside edges, frames 1 and 10, although with a super it can be quite a good idea to have 9 frames in there, nicely spaced, because it just encourages them to make deeper cells on those frames. So let's say you're looking at frames 1 and 9 can be empty, and frames 4 to 6 or 7 can be filled up and capped. So if you rotate those outer frames into the center, you center to the outer, then you're making sure that you're getting full width of the box without having to simply have 10 boxes high on your hive. When you do put a super on, take a couple of frames from your existing super and introduce them up into your new, along with the bees on them, and put a couple of empty frames, or either drawn out or bare foundation into your lower box. That thing of introducing bees and honey into your new super will simply get movement from existing area up into the new super a lot faster, acceptance, and they'll start using that super and storing in there much quicker. Your new super can be either on top or underneath your existing. So on top has the advantage that you can simply lift your lid off and check much more easily as to how quickly they're filling it up and whether you're wanting to rotate center to outer, outer to center, that type of activity. But the bees have got a little bit further to go to carry the honey up. Personally, I don't think that's much of an issue in a good strong hive. Under supering means they don't need to go as far, but you're going to want to then lift your top box off and see how that newer box underneath the top box is going in terms of being filled up, and do I need to add another super on yet? So different alternatives there, and entirely up to you. Try one hive one way, the other hive the different, and see which way works best for you. Ventilation and cooling can be quite crucial at this time of year. I do recommend a mesh or a ventilated base. It not only has the opportunity to bring cooler air up and into the hive, but it also means that any debris and dead varroa mites or mites which have been cleaned off the bees as a result of grooming simply drop straight out the bottom of the hive and can't have the opportunity to attach themselves to a returning worker bee as she's coming back in and walking over a solid base. A hive stand in combination with a ventilated base can be quite good, lifts it up off the ground and will increase in the airflow even more. You can have a ventilated hive mat. So on your top mat, underneath your lid, you can cut a hole into that and then put a piece of mesh, either stainless steel or something else fairly fine. This then allows warm air to rise up through the hive and actually escape out of the hive. If you're going to use a ventilated hive mat, I would possibly suggest having a spacer up above it as well, because otherwise, of course, if you're using a tin or galvanized lid, then the heat of the sun coming onto that lid might actually mean warmth coming down into the hive, whereas if you have a spacer, so something say about 3 or 4 inches high, just a surround, the bees can't get up and into that space, so they're not going to drop comb to it, but it allows room for the air, the warm air, to move up out of the hive and into that attic space. Can be a good idea also to just, when you're reassembling your hive, just put a matchstick at the corner of the supers. So this allows just 1 or 2 millimeters of space. This is enough for a bit of air to move in and out of the hive and warm air to escape, but it's not enough for either bees or wasps, robbers, or anything else like that to get into the hive and cause a problem in there. So a matchstick at a couple of the corners is good. And of course, a supply of water nearby. The general recommendation I've seen is don't put that water immediately next to the hive, but instead put it a few meters away. Rainwater preferred to tap. Like so many things, the bees actually like something with a bit of smell, and so a bit of grotty old rainwater that's been sitting out there for a while is absolutely a gin and tonic to them, with ice and a slice. They love it, and they much prefer that to nice, clean, fresh tap water which has been chlorinated. Remembering, if you're providing a supply of water, then either stones or bracken or something like that into it. I've just got a couple of good-sized planter plastic bases. You can get them at a garden centre. Only cost $1 or $2, but anything like that. And then fill them up with stones, put water into it, and that provides a water source for the bees. Remembering, of course, the old adage, each new bee requires a cell of nectar, honey, a cell of pollen, and a cell of water. So water year-round, not just during summer, but year-round can be a good idea. Don't be worried if you see the photograph on the right and all the bees out front and hanging off the front of the hive like that. I've had photos being sent to me over the past few weeks with hives looking similar to that, and people worried that their hive was about to swarm. This is just normal behaviour on a good hot afternoon. I find about 3 o'clock in the afternoon, there's hundreds of bees climbing over the front of my hive, similar to that photograph on the right there. And this is simply the girls maintaining a good balance of temperature. And simply with that many bees inside the hive, it can get too warm for the brood, so they come out and hang out outside. So it's not an indicator of a problem or swarming or anything else like that. The girls are simply maintaining temperature within there. And holidays. They always seem to want to do something just as we're about to go away on holiday or 2 days after we have gone. So go into your hive and have a good look at it before you go away on holiday. Provide additional space. During a good nectar flow, a good strong hive can fill a 3/4 box within about 3 to 4 weeks. And that's draw comb out, fill it up and cap it. So if you're going away down to Coromandel or up north to the beach or something else like that, and you're going to be away for 3 weeks, Christmas and into the middle of January, then provide an additional box onto your hive to allow for the girls to have filled it up while you're away, and make sure that they have plenty of space there. Top up your water supply. Do an inspection, and this means also going down into your brood area and making sure that your hive isn't going to swarm. The last thing any of us want is to go away on holiday, find that a hive has swarmed while we've been away, and then, of course, we're not there to oversee the requeening of the hive, making sure that it's gone properly, everything like that. And we end up worrying while we're on holiday rather than lying on the beach and having a good relaxed time. So go and do an inspection of your hive before you go on holiday and make sure that it's set up for you to be away. Let a neighbour know that you're going to be going, and perhaps give them a contact of another beekeeper, a friend, somebody in your group, something like that, who they could give a call to if something does happen while you're away, and maybe arrange with that person to even come in, do the occasional check on your hive, something like that, to just keep an eye on things and check on them while you're away. So all of these, prepare in advance, know where your hive's up to and so forth, and that way you can go away on holiday, have a really good relaxed time and not be worrying about your girls, and they can be working flat out and furiously and present you with lots and lots of honey for extraction when you come back from holiday. Okay, that's all I wanted to say on that one.
One of the advantages of belonging to Auckland Beekeepers Club is being able to borrow, hire some extraction equipment. The details are in the journal, in the back of the journal, about how you can borrow the extractors. There's 2 4-frame extractors and a honey press as well. The 2 people who are looking after them and arranging the rental is Alan Harwood, he's in West Auckland, and Bruce Ranby, who's on the North Shore. Their phone numbers are in the journal, so phone them up and arrange a certain day that you want to hire it. You will have to pick it up yourself and return it. It's best to book in advance because there's only so many weekends during the extraction season. As you can see, they're quite large, so you need a decent sized car to put it in, or a trailer or a station wagon. It's only $25 per day to hire it, but there is a $50 bond to pay as well, and the bond will be returned when the equipment is all returned and it's clean. If it's not clean, then it will have to be cleaned, and someone will have to pay, and we have to pay someone to clean it, so you could lose your bond if it's not cleaned properly. The extractor is provided with a soft brush to help with cleaning. And when you clean it, use cold water and dishwashing liquid to clean it thoroughly, and then rinse it off, and then make sure it's dry so that it doesn't rust or any of the parts get tarnished. Make sure you clean off all the honey and the propolis and wax that might be on it. Don't use a water blaster, because that could damage the seals or parts of it. So there's also supplied with it, there will be a spanner which will be help to undo these two nuts here so that you can take the lid off to help with cleaning. Make sure that you don't lose any parts, especially the bearings or the spring that's in there. I'll show you that when we come to the cleaning part. Also, you will usually be supplied with the, these two plastic containers for uncapping. So there is, the first one is a sturdy plastic tub, and the other one fits in top, but turn it round and it sits on top of it like this. There's a lid and there's holes in the base. So when you are taking the caps off the honey and the caps and the wax goes into here, it will drain through to the bottom one. This one just has a bit of mesh, but I think there's the other ones are slightly better than that. Then there is usually a frame to put this on, and then you can put that on there when you cut the caps off before you put it into the spinner. So the most important thing to remember, or amongst the most important things to remember, is when you bring your honey supers in, don't bring bees in. Make sure you get rid of all the bees, because even a couple of bees that you bring inside, they will die, and usually by stinging you. So don't bring any bees in, and it goes without saying, do it inside. Do it in the kitchen, do it in the shed, do it somewhere inside, because especially if you've got bees in your hives in your garden, you will end up with bees, hundreds and hundreds of bees. If you leave the window open, you'll end up with bees. So make sure that it's bee proof where you're doing it. So I'll just show you the parts to it. There's two nuts and there is some washers. Don't lose any of the parts, otherwise they will have to be replaced at your expense. So, and then the lid just lifts off. The lid is hinged, and then there's the handle that turns the basket. Now with the handle, you might be able to see that it locks in. So when it's pulled outwards, it just spins. But when you lock it in and turn it, it will turn the pivot. So bear that in mind if you're turning it and it's not spinning. And then inside there's the basket, which is what spins round. It goes in one way, it goes in this way, because on the bottom there is the racks that hold the frames. Now these are designed for full depth frames, so they fit in quite nicely, full depth frames. But most people will have 3/4 depth supers, and when you put them in, it doesn't necessarily sit in properly. So you just bear that in mind, and it might have to go on a slight angle to sit in properly. It won't fit straight on, it could go through. So you just need to make sure that you have some way of doing it so it's on a slight angle. And then when you're putting it in, you must make sure it's balanced, because if there's too much weight on one side, then when it spins it will wobble. So you can have 2 frames or 4 frames, but make sure that it's an even amount and they're balanced. So if there's one frame that's full of honey, really thick, don't put it next to one that's got less, it's a lot thinner, otherwise it will be unbalanced. And also be aware of, don't spin if it's uncapped, if there's lots of just nectar in there, and otherwise there'll be too much moisture and the honey will ferment. The honey does need to be less than 18% water. So if it's too much water in there, it's too much uncapped honey, then it will ferment. but you can have some uncapped honey, especially if it's very thick uncapped honey. So a rule of thumb would be to shake it, and if it comes out, then you know that it's quite runny and it's not been cured or ripened. But if it's uncapped and it doesn't come out, then you know that that is ripened anyway, so that will be fine to extract. So then the basket goes in and it sits on the pivot in the centre. And then, so this is interesting to note that this is how you'd clean it. You'd dismantle it like this, and then you'd clean it out and wash it out and make sure it's dry, and then reassemble it. So make sure the pivot clicks into place. Like so. And here is the locking nut. So that goes in and up like this, and then the washer goes on top, and then the nut goes on to hold it in place. So that is how you reconnect it. So that goes in and up, and that goes onto the metal frame. Washer, nut, same again on this side. Make sure that it's still sitting on the pivot. So then, when you use it, you click it in and turn it, and it spins. And when the handle comes out, it's still spinning, but the handle's not engaged. So you push it in, and the handle engages. So when you spin it, start off slowly at first and slowly build up the speed, but you do not need to go as fast as you possibly can. There is a reason for that. That is because if you have a wooden frame with wax foundation, even if it's wired, and you spin it too fast, the wax will just push off the frame and it will damage the foundation, and you'll end up with the wax inside and nothing on the frame. With plastic frame, you don't have to worry so much because that's solid, so that when you spin it, it won't push through. In fact, a little trick is to, when you spin it, is to spin the wooden frame just a little bit around a few times so that some of the honey comes out, and then that takes away the weight from this side. Turn it over so then that there's less weight on the inside, because when you're spinning it, it comes off the outside. So then there's less weight on this side, so you spin that round and take the honey off that side, turn it round again, and then the first side, you spin it and take the honey off that side. And with the plastic frame, you can just put it in there and spin it round and take all the honey off that side, and then take it off this side. Bear in mind that it should always go with the bottom facing the direction of spin, because the frame, the cells are slightly angled. They're angled upwards. So if you were to spin it the other way, what it would do it would rip off the cells and the cone. So always do it so that it's facing, the bottom is facing the direction of spin, so that it's easier for it to come out. Okay, so when you have the honey, when you're uncapping, my favourite way is with hot knives. I have a hot plate, electric hot plate, which I just put a saucepan filled full of water, and then I have a few knives in here, and I will dry off the knife, and then the heat of the knife is still hot, so I will just cut off the the capping and then just swap over and get another knife. Make sure it's nice and dry because you don't want to add any moisture to the honey. So then you would just, in a sawing motion, cut the caps off and then put it in the spinner. There are electric knives or steam heated knives which are probably more effective, but they are more expensive. Also, you could use a capping tool, which is just lots of prongs on it with a handle. And after a while, you could become quite adept at taking off the cappings with this. But also, this is quite good because if the cells are quite low and the knife running along the edge of the frame misses some of the caps, this you can just take out, usually in the corners or on the edges. You can use this as an extra thing to make sure they're all uncapped. So if it's not capped, then you're not going to be able to extract the honey from it. So here you'll notice that there is a tap underneath. It's called a honey gate, and it flips over and there's a screw to lock it in place and there's a seal to stop it coming out. So you can only extract so much because there's about that much before the basket hits the level of the honey. So you will usually need to put a bucket underneath there and then with the gate open so that when you spin it, it goes straight into the bucket. Use a food grade bucket that hasn't been used for anything else. It's quite useful to install a honey gate or a tap on the bucket so that when you want to fill your jars, it's quite easy to fill your jars. At this stage, it'd be good to filter the honey so that you get all the bits of bee out, wax and any other bits. This is quite good, available from most suppliers. It's a two-part metal strainer, a coarse sieve at the top and a finer sieve at the bottom. So that just sits in there like that and strains the honey as it goes through. I think slightly finer is this nylon net bag, which is I think a jelly bag, or you buy it from the suppliers or AliExpress. Put that on there and it will strain the honey that goes through. So you don't want it too fine because otherwise it will take out the pollen and some of the the good bits in the honey. So then obviously make sure that when you're spinning it, it's nice and balanced. Start off slow, click it in. Start off slow, just get some of the honey coming out, start off slow. And then if you have wooden frames, then you would take them out, turn them round, and then spin it some more. Just gently spin it. You don't need to just go as fast as you can to get it through breaking the cone. So just gently, and you could hurt your wrist, so that's one of the good things about unlocking. So you just gently spin it like this and then turn over the frames to spin the outside, and then when you open it up and have a look, make sure all the honey is off and then put it back in the box to and then you can consider, these are called, when the honey's been extracted, these are called wets, and then there is a way of putting the wets back on the hive so that the bees will clean them off. Also, we have, some of the extractors have this little frame. It's got holes in there for the feet. So this goes underneath. The feet sit in. And it just gives it a bit more stability for when you are spinning. So it just gives it a bit more balance. And also, when you're draining it out and there's the last bit left, you can raise up the back and tilt it so the honey will come out. Now the honey press, this is my honey press. It's similar to the club one, but the club one's slightly different. So but it's the same principle. This is useful if you have a top bar hive or you only have a few frames to do. This has got a bottom plate to put in here and a spout. So when the honey is crushed, the honeycomb is crushed in here, the honey will come out here. So put a bucket underneath with a filter, and then what you do is there's several ways of getting out. You either, if it's a plastic frame, you just with like a paint scraper or something like that, or a hive tool, you just scrape off all of the comb, or if it's a wooden frame, just snip the wire and then get a knife and just cut round, and then dump that into some muslin or a cloth bag, and you put that inside there. Then you put the top plate on, which has a handle for pulling it up and a place for the screw to go into, and then you just squish it down, and then when it's as far as you can go, leave it for a bit, 10 minutes or so, and you'll find that a lot of the honey has come out. Keep going a bit further, and then a bit further 10 minutes later, and just keep going until you've squished out as much as you can. I find this is quite good for if some of the frames have manuka honey in and they won't spin out. No matter how much you spin it, the manuka or kanuka won't come out because it's very thixotropic. So I just cut it out and put it in here. And then also with this, you leave it for a while and it's still coming out, then you can just put a something under the back so it'll pour out into the bucket. So, and then sometimes when it's peak time, there might be someone, if you borrow it on the Saturday, someone might have it on the Sunday. So you'll have to arrange between the two of you for pickup or delivery. So if you've borrowed it, respect everyone and make sure that it's all the parts, everything that you've borrowed is returned. It's returned in a timely manner, but most importantly, it's clean. Make sure it is very, very clean. Cold water, dishwashing detergent and a soft brush, and clean it thoroughly, drain it, rinse it well. Make sure you get off all the honey and any propolis or wax, because that could contain spores that you wouldn't want to contaminate stuff with. So just and make sure that's returned, and yeah, enjoy your honey.
Okay, that's great. Thanks for that, Ken. There's just a couple of things I'm going to comment about on that one. First up, there are 3 club extractors which are available for hire, not 2. Alan Harwood has one for West Auckland, Bruce Ranby, as you say, for the North Shore, and I have the third, or will have the third, I'll get it in a week or two's time, for myself for the Central region. So 3 club extractors. Also a note, as far as I'm aware, none of us actually rent out the uncapping boxes as well as the extractor. For myself, it's really just the extractor. If you want a spanner to go with it for the nuts and things like that, that's absolutely fine. But certainly I don't, for all the club hire of extractors, I don't lend out the uncapping boxes and things like that. I expect people to provide their own for those ones, and happy to give advice on what works and things like that. Another small one there, I would say, in that center, so you've got the basket which lifts out and then goes back in. Underneath that basket, and where the basket fits into the base of the extractor, there is a ball bearing. And a number of times people, of course, take the extractor out after they're finished, wash it out, pour everything out, and then they turn the extractor upside down to get all the water out from inside, which is perfectly valid and good. But the ball bearing comes out because it's just sitting there at the base of the extractor. It's not held in by anything, so they can lose the ball bearing. And if that happens, I'm going to need to ask for a few dollars to get a replacement. So if you can make sure that you don't lose the ball bearing when that comes in there. Also, when you're, as Ken showed about a little gear mechanism on the winder, so you press that in and then you start winding, it engages the gear, and then that turns the basket round and round. And then, of course, as soon as you stop winding, it's spring-loaded, so the handle pops out slightly and disengages from the gear, but the basket keeps on going round because it's got the weight in it and everything like that. Don't try and press that handle back in and then try and slow it down using the handle, because you'll find that, of course, as soon as you engage that gear, that handle flicks around, catches you in the back of the wrist, and will leave a nasty bruise, if nothing worse. Actually, just grab hold of what is basically the axle that runs from the handle through to the center pivot there, and then just put your hand around that and grab hold of it, and that'll slow the basket down from spinning, which then means that you can open up your two top flaps, turn your frames around, as Ken says, and do things like that. I provide a little information sheet when people borrow the extractor from me, which has got this sort of information on it as well, just as a reminder for when you're going through. But that's an excellent presentation. Thank you, Ken, for that. I don't think that we've got any questions or anything in there. We're 12:30 now. We've been going for a couple of hours. It's still a grotty day outside, but I'm sure you're all probably wanting to go and grab lunch and carry on with other things to do. So unless there's anything pressing, or Dave Hendricks, or do you have any comments that you want to add into there?
No, I'm good. Thanks.
All good. Righty-ho. Thank you very much indeed for everybody who came along today. Much appreciated. Thank you to Heather Hendrickson for an excellent and fascinating talk, and for Ken for a presentation on those two videos he's made. As I say, December meeting, the 11th, looking to be in person. Put a ring around it in your calendar, save that date, and I really look forward to catching up with you again, seeing you, and finally being able to do it in person, and yeah, have a chat and catch up on all the news. In the meantime, be good, keep your bees healthy, and yeah, take care. Cheers.